Alright guys, for this section, what we're going to do is we're going to look at the genetics problem packet, and we're just going to identify some issues that some people had and just kind of do a little refresher on this particular worksheet. So for the first one here, we're going to look at the alleles in this second section. A few people had trouble with this. When you review the instructions for it, it really does kind of explain itself. If you understand what the words dominant, trait, and recessive mean. So for this, you're going to look at the characteristics. So for number one, we're going to look at smooth and wrinkled. And the instructions say that you're to use the first letter of the dominant allele, and the dominant trait is written first. So smooth is our dominant trait. So we would use a capital S to refer to the smooth characteristic, or the allele that codes for smoothness. And then we would use a lowercase s to indicate wrinkled. So our key would look like this. A capital S would be smooth, and a lowercase s would be wrinkled. And you're just going to go ahead and do this for all of these. So, for example, for number 5, I would suggest using a capital Y, and then for the green stem, using a lowercase y. Okay, then when we go ahead and look at come down here and look at setting up a monohybrid cross. Some people had trouble with this as well. I'll just go through it real quickly. Possible genotypes that you can have. All you need to do is pick one of these up here that we had just done, and I'm going to use dimples and no dimples. So I'd use a capital D for dimples and a lowercase d for no dimples. And we're going to go ahead and list the possible genotypes here on the key. So possible genotypes, this is the combination of alleles that this individual could have. So capital D would code for the phenotype of dimples. Capital D, lowercase d, would code for the characteristic of dimples as well. And this is simply because dimples is the dominant characteristic. And the last one, we'd have lowercase d, and that is an individual that has no dimples. Okay, now the cross, what I would suggest doing is we would do a cross between a heterozygous individual and a heterozygous individual. So we go ahead and fill out these alleles for mom and the alleles for dad. And some of you probably noticed that mom and dad are switched on this worksheet. I had mentioned to you guys that it doesn't really matter. In fact, most textbooks put dad on top and mom on the side. But really, I have no preference on how you do that. So we go ahead and fill in our Punnett square. And our Punnett square should look like this. And then it's going to ask for genotype percentage and phenotype percentage. So all we're doing is we're listing the percentage of the individuals that are capital D, lowercase d, or lowercase d. So if I were going to use the correct terminology, I would say the individuals that are homozygous dominant, the individuals that are heterozygous, and the individuals that are homozygous recessive. So how many individuals on this Punnett square right here are homozygous dominant? When we take a look at the Punnett square, we notice that this is the only one, capital D, that is homozygous dominant. So we would say capital D, are homozygous dominant, and we would list 25%. Then for the next one, capital D, lowercase d, or heterozygous, there is 1, 2. So there's 2 out of 4, that's 50%. And the last one, lowercase d, there's only 1 out of 4, so that again is 25%. Always check yourself by adding these numbers up, and they should add up to 100%. Okay. Now for phenotypic ratio, or percentage I should say, all we're going to look at for phenotypic percentage is we're going to count how many of the individuals have dimples and how many don't. Remember, for phenotype, the PH, I'll have you guys remember that that stands for physical. Think of physical characteristics. It's what you can see. Okay, so we can see dimples. We're going to look at those and we're going to say, okay, capital D, does that individual have dimples? Yes, that's 1. Capital D, lowercase d, does that individual have dimples? Yes, that's 2. capital D, again, yes, that's 3. Lowercase d, no, that individual does not. That's 3 out of 4. What percentage is that? It's 75%. So when we put this on our answer key, what we're going to do is we're going to write the word dimples, and then we're going to put 75%. And that leaves 25% with no dimples. For the genotypic ratio, we just count how many individuals are homozygous dominant, how many are heterozygous, and how many are homozygous recessive. So when we count those up, we find that 1 is homozygous dominant, right here, capital D, capital D. 2 are heterozygous, and 1 is homozygous recessive. That gives us a genotypic ratio of 1 to 2 to 1. For phenotypic ratio, we're using the ratio of how many have dimples to how many don't have dimples. 1, 2, 3 have dimples. 1 does not. 3 to 1 is our ratio. All right, moving along to the next page. If we take a look at the next page, we look at this on the complete dominance. We can take a look at free earlobes real quickly. And what we find for free earlobes is that free earlobes are dominant to attached earlobes. Okay, so I'm going to use capital F for free and a lowercase f for not free. Okay, you're going to make a key for the possible genotypes with the corresponding phenotypes. So all you have to do is write the different possibilities. An individual could be homozygous dominant, an individual could be heterozygous, or an individual could be homozygous recessive. This person is going to have free earlobes, this person is going to have free earlobes, and this person is going to not have free earlobes. And erase this. Now, if we are going to go ahead and make a cross, that just means that you're going to make a Punnett square. So I make my Punnett square, and we're going to have a pure, free, earlobed man. And what that means is he's homozygous for free earlobes. So he's going to have capital F, and the person with an attached ear is going to have to be lowercase f, lowercase f. When we fill in our Punnett square, we notice that all of the offspring must be heterozygous. So then when I ask you for a genotypic ratio, or phenotypic ratio, or phenotypic percentage, we're going to come up with virtually the same answers. For the genotypic ratio, we have 100% or 1 heterozygous. For the phenotypic percentage, we have 100% free earlobes. And then for E, the phenotypic ratio, we just again have 1, and then we list the phenotype. So that's free. And that's it for number 3. So as we scroll down and look at the next page, what we're going to see is that we have some concerns that people had with, let's say, number 7. So let's look at number 7. Draw a cross between a circle star-eyed and a circle-eyed smiley. Well, this is an explanation for this incomplete dominance or this codominance. So we're going to have to make a key first. And so if an individual is going to have what we would say circle star eyes, then let's go ahead and use let's use E for eyes and superscripted C for circle, and then again E, and then a superscripted S for star. Now this individual would have circle star, and we're going to cross that individual with one that has circle eyes. So that would be EC, EC. Now when I go ahead and draw out my Punnett square, I'm going to take these alleles and I'm going to put them in the Punnett square just like I did with all of the others. And it's the same concept. EC, 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 ES, ECEC, and ECES. Now we just have to answer some basic questions about it. What percentage of the offspring are circle-eyed? Well, this individual right here is circle-eyed. This one is not. This one is circle-eyed. This one is not. That's 2 out of 4. So you should have, for letter A, 50%. For letter B, what percentage of the offspring are circle star-eyed? Well, obviously, this one is not circle star-eyed, and this one is not circle star-eyed. And when we look at these, they have an ECES. That tells us that they are circle star. And so we have 2 out of 4. So your answer for B should be 50%.